Hi, my name is Nicholas Correll. I'm an associate professor at the University of Colorado at Boulder. And I will talk about bronze, iron, plastic age. How will the next material revolution look like? Thank you very much for inviting me to speak at FNANO. I'm very fond of the DNA self-assembly community. Although I work on a completely different scale in robotics, I hope though to have some ideas that I can share with you and that you might find inspirational, in particular, in a way to work together in the future. So I really like the idea of having cellular materials, and cellular materials are actually very common in nature. Every material that nature produces is cellular. Here I have a couple of samples of cellular materials that people like to work with for thousands of years. So you see wood, you see leather, you see bone. And in the bottom row, I have some mineralized versions of those cellular materials. So the cells have been grown and then they mineralized to become sturdy and hard. So there's horn and ivory and muscles. But I think much more interesting than cellular materials that can just grow, and you have an abundance of those, are living cellular materials. So think about living wood that is able to repair itself, strengthen itself, grow in a certain direction to follow the sun. Think about living skin that is able to sense touch. Think about bone, which is essentially a power plant in the body that can generate white blood cells and measure the strain that you impose and then adapt accordingly. So when you think about these living cellular materials, the most exciting one on planet Earth is probably aside of the human brain, the octopus. So here's a fancy video, and I apologize, I don't speak Japanese and don't find the original version because it has been pirated so many times. Here you see an octopus that is trapped in a jar. And you might know that the octopus has 2/3 of its neurons in the arms. So even if you cut off the arm is able to, for example, detect food and move it where it thinks that the mouth of the octopus is. Now what happens here is the octopus trapped in that jar and starts getting out by itself. So somehow coordinating millions of neuron cells, muscle cells, sensing cells, and so on, to accomplish some feat. Let's wait for this real quick until you see now it happened. The octopus has somehow opened the jar and now comes out in its full beauty. Now, if you think about how to build such a thing, the way I think about it is that you could have little cells, each of which have sensing, actuation, computation, and communication therein. And now you could embody these cells, or embed these cells, sorry, in some kind of smart polymer and create bones or muscles, brain, and so on to build more complex robotic systems, for example. And so in addition to making those cells and thinking about ways to integrate them with actuation or sensing, you also need to think about how to coordinate them. So in a muscle, all the muscle cells are acting in concert. In a skin cell, some cells sense something and then send the information somewhere else, and so on. I think one good way to think about this is as a global to local problem. I have a certain global behavior that I want, but I can only control the local constituents. And so how do I solve that? There's a lot of related work, especially from my perspective. This is absolutely not at the nanoscale, but that's how I like to spend my time. There has been the amorphous computing paradigm from MIT that then turned into the paintable, or had some subset of it was the paintable computer idea. And these guys put little robots on a wall and made them create the pattern A in concert. So the idea was we could make them eventually small enough, they get powered somehow from the environment, they talk to each other, and you have some kind of smart paint that you can just paint on any surface. There is programmable matter from CMU. Here the idea was that such smart paint elements could also move and self-configure. In general, swarm robotics is concerned with the idea on how we can form patterns, how can we, assemble ourselves in a certain shape. I think that's where the relationship to DNA self-assembly is most obvious. People have started exploring these things in bacteria. How can I program bacteria to do certain things, like exhibit a certain pattern in response to a certain chemical? That was work at Princeton, now at MIT in the 2000s. People have built very, very small swarm robots. So the iSwarm project is maybe the most notable in Europe. Here you see a little swarm of robots that is powered by solar cells. They can communicate with infrared to some extent. The whole field of modular robotics, of course, is concerned with these kind of ideas. And I personally try to make smart composites that embed many, many computers and create some interesting behavior like shape change or detecting touch or creating camouflage. And so my quest is to make these things smaller and essentially create a smart material system that you could buy at Home Depot, and which can either change its shape or can help, you can put it over something and it provides that thing with sensing or with camouflage ability, just to name one example. So in order to have this discussion with you, I think we should just agree on what computation is. There's nothing new on the slide, but I like just to set the stage that way. So I think about computation as a universal theoretical construct that can be implemented in any way you want. In this case, what I'm showing is a feedback control mechanism that is implemented in fully digital ways in analog electronics and eventually in completely mechanism. I would argue that these systems are completely functional equivalent. So what you see to the left is called the Watts governor. It's a steam engine regulator. The idea is that you have a flywheel and two balls that will spin up due to the centrifugal force when the steam engine starts spinning. And as the steam engine spins faster and faster, there's a mechanism that the lifting balls will trigger to actually regulate the inlet to the steam engine and thereby controls it. The same thing here is an op-amp circuit where you can set the set value of the voltage that you want at the output using two resistors. And finally, you could write code that does the same thing, in which you would measure an actual value. You subtract the desired one, which is your error, and then your output is essentially a bias term plus some gain times the error you got. So if the error is 0, you have no output or just a bias term. If the error gets very large, the output goes in one way, and if it gets very small, the output goes the other. So to me, computation is really universal in the senses that you can replace one with the other. You can build systems that mix these things, that use mechanism design, that use electronics or code. And again, the code you could implement in all sorts of ways. You could implement it with transistors, you could implement it with polymer electronics, or you could even implement it with DNA computing. So DNA computing is what most people here do. I don't have to explain too much of it. I just want to ground it so you understand how I think about it. To me, DNA is a very big memory that is in every cell in the human body or in any actual natural material. And interestingly, every cell not only carries its own blueprint, but the entire blueprint for the system that it's in. So if you go to an engineer and tell him that, or her, hey, make me a system in which you have microscale components, each of which includes the entire blueprint, they would probably think you're crazy. Now, how crazy is it that nature does it that way? I don't know, but I think it's a very good motivation to actually not completely discard that idea. So first it's memory, second is computation, and then Adelman came up with this idea with the Hamiltonian path problem or the traveling salesman problem. And I think it's a very nice example to see that solving a computational problem using DNA is essentially a self-assembly problem. So this very canonical problem, the idea is that you have a set of cities. Let's say you have 10 cities. Here we have Miami and New York, and you would have 100 routes that are possible. So you could go from Miami to New York, you could go from Miami to Boston, you could go from Miami to Denver, and so on. And so when you encode this, the DNA, the amino acids you choose are actually complements to the end of the Miami code. And here, these are the beginning of the complements of the New York code. So this GCC will hook up with CGA and TAC will hook up to ATG. Now, let's say you have a jar with like 1 million of these cities and a lot more of these routes. You shake it up well, it's very likely that you might get a route that encodes all of them and is actually the shortest one. And what you can do is you can then look at the DNA and extract it and take the one that you like, which has the right length and starts at the beginning and ends at the end. And there you have your solution. So now that I think I convinced, or we are on the same page that computation is assembly and the other way around, people have gotten very good at assembling anything they want. So here's an example from the Wyss Institute where somebody has assembled all sorts of characters and letters and symbols and so on. So I think this field is pretty mature now. We can assemble whatever we want. So this was all at the nanoscale. If I have to compute, I actually am very fond of microscale computation. And also this field is very advanced. So, you know, the neuron in the brain is also very great memory. We know that because we know a lot of things. And so it works very well. We understand very well how to reduce it into a computational system that we can analyze, also known as the artificial neural network. We all know it's not how the brain really works, but at least it's some very good way to get very, very impressive computations done. And we are able to implement this in silicon. So we can implement it in silicon, we can implement it in polymer, we can implement it in novel neuromorphic, transistors or neuromorphic cells that allow to mimic the brain operation more closely. Now the problem with this is it's really microscale. So if I want to build a system of cells to make my materials, I don't think I get very far with a microscale. So here's my proposal to you. I think what we should do is we should think about ways to combine nanoscale and microscale computing. And in particular, there is a lot of room at the bottom, as Richard Feynman said so famously. And I think that's true. If you can move any computing that I would want to do in creating an interesting cellular system, if you're able to actually move that into the nanoscale, you can do almost whatever you want. There's so much space in a microscopic cell to house so many nanoscale components that I think it would be possible to create very powerful cellular systems. Now let me elaborate what I actually mean, because otherwise. So the way to think about it, in my opinion, is about the interfaces to sensing and actuation. So if you think about microscale computing, which is very well understood, there is almost, you all know you can compute anything you want, but we also have MEMS devices that allow us to provide actuation at the microscale. We can also emit any electromagnetic spectrum that we want, also known as light or heat in the infrared spectrum. And at the same time, we can sense almost anything. So we can sense molecules in gas, we can sense light, we can sense heat. And now if I have to build a muscle cell or something like this, what am I going to take? Am I going to take a microscale actuator or microscale sensor? Or am I going to take a nanoscale sensor and actuator? And if I can do that, then I probably be able to do that at a much smaller scale than I could do at the microscale. So I think we are on the same page that nanoscale computing is very well understood. we understand the DNA, we understand how we can lock and unlock processes of computational nature. There's even some actuation that we know how to do. So very famous is the DNA walker that has been studied to great extent. We also have an inkling of understanding, or actually probably more than an inkling, on how to make proteins. So if you have something that can be actuated by proteins, you could actually make a muscle, and you can use DNA to lock and unlock this, this activation by having the RNA create proteins with a ribosome. There is some research on DNA emitting light, being fluorescent. I'm not sure how much research there is on actually using DNA as a sensor. So can I sense heat? Can I sense light? Probably yes, because I can use it to trigger certain things in the self-assembly process. But there may be other things that I could do. Maybe I could self-assemble a structure that is able to recognize certain patterns. So this is my proposal, to think about DNA self-assembly as actually the operating system for microscale cellular engineered systems, which, by the way, are very similar to how nature already operates. But so it's not really my idea, but I think it's still something that maybe doesn't enjoy too much attention right now. So the other thing that is important is communication. If you have a heart or a muscle, then these things are usually coordinated. And communication is something that we can do really well. And we can do it at very high speed. So we can send, for example, electromagnetic waves over a wire or light and have these cells, for example, synchronized or something like this. But what is possible on the nanoscale? Of course, we could just split off RNA and send it over to another cell using some kind of substrate. This will be probably a very slow, diffusive process, but still, it's something that we should think about. For example, when you do camouflage, why would you need expensive, expensive in terms of size, microscale electronics to do that? Why not just use the diffusion processes that camouflage relies on, and send RNA to neighboring cells, which there unlocks a process that makes them change their appearance, and again, send RNA to their neighbors. So this is stuff that I like to think about computationally, but it could also be done, realized in a material. So I came up with a to-do list to do that. So the first thing we have to do is we have to understand computation and self-assembly at the nanoscale. I think we do. This is a very mature field. People know a lot of things about that. But at that point, we probably don't have a survey of what actually can be sensed at the nanoscale and what can be actuated. So if we had surveys like that and would sit down as a community and think about that, we might be actually able to design novel micro-nano interfaces. And so if you had those micro-nano interfaces, you could actually have the DNA computing do something in the real world. In the micro and mesoscale. And at the same time, you could control the DNA self-assembly process from the microscale. Some things are very simple. You might have a little heater, you might have a light emitter that triggers certain processes. Others, I think, require more research. So once we have those things, I think we can start deriving mathematical reductions for cells that have such a nanoscale OS. And here, I don't think we have to wait until people have actually built them, but we can just think about what could be possible by doing a survey and thinking about these things, and then have other people start thinking about what those cells could do if they had those capabilities. And in particular, we could think about what kind of global pattern matching, and so that's, for example, recognizing something in a bunch of cells, and pattern generation, such as camouflage or motion, is possible. So for discussion, I think I'm very curious how many distinct pre-made cell types you would actually need to build anything interesting. So in my own research, we have built something with the friends from Fem2ST in France to implement Tetris on a discrete set of cells that each can light up in a different color. That's what you see here, what somebody holds. And there's a tetromino coming down, and you can like twist this thing, and the tetromino will then rotate and fall and then into this gap, and then the cell, the row will disappear. And so all of this is happening with a distributed algorithm. But you clearly need more cells if you want to build something more complicated, maybe a little animal that can walk or something. And so what I find very surprising is that the human, that is very complex, and the most complex thing that we know on the planet, actually is only made of 200 different cell types. And here for your reference, I have a little log plot on the different species and how many cell types they have. And so for some crazy reason, animals that are much less complicated than us have more different cells. So it's something we really don't understand. And I don't even know how to go about it. But one way to think about it might be to think about the basic primitives that an interesting, in quotes, system needs, such as synchronization, consensus, localizing of the cells within the body, and so on. And maybe there's something, some formalism that we can reason about, something like NP in computer science, or NP completeness. Can we, once we have, what are the minimum primitives that we need to create a system that has certain computational power, certain sensing power, and certain actuation power? So let's say something that can run, avoid obstacles at a certain rate. So I have difficulties even defining what the benchmark for that would be, but I think it's worth thinking about. So with that, I want to conclude. I think it's worth reminding ourselves about Feynman's famous quote. There's plenty of room at the bottom. If we really want to build very small things, I think we should reactivate that. I mean, me personally, the DNA self-assembly ideas, and think about what they could do for us if we want to create material systems that have cells that are at a reasonable small scale. I think that the DNA computation field really needs an unlocking by focusing on microscale interfaces. So if you had a way to steer and control a traveling salesman problem and then actually read it out, that's when you actually get to the DNA computer. I don't think it's sufficient to just collect the DNA in PCR and then look at them in a gel. And finally, I think it's time to think about theoretical reductions that could inform, that are informed by current trends. So let's say you find something that can be actuated by a DNA computation step, then I think it's time to think about what could we do with that, and then once we have a compelling demonstrator, drive actually the experimental science in biology and chemistry to make us these things. So I was really hoping to come to Snowbird to discuss and actually get input from you guys. This is not possible, and I'm not sure how it's gonna work. I uploaded this to YouTube. I would really appreciate maybe we can get a discussion going with the commentary section in YouTube. Otherwise, please email me, nicholas.correll@colorado.edu. And I hope I see you all again very, very soon. Bye-bye.